All right, guys, you ready for this? It's going to be awesome.
What did you say you were?
We're doing the cell cycle. So we're going to cover the cell, mitosis, mitotic division, and we're covering meiosis. So pretty much, how's the nucleus divide? How's the cell divide?
But it's 9 and 10, correct?
Oh, chapters? Yeah, I believe so.
So can we do just 9?
No. We're going to do the whole thing because they don't make sense without each other.
Okay, let's be really speedy. Let's do
We're going to do this, and we've got at least, 2.5 hours that we can do this in. So we're set.
2 hours.
2 hours? We'll see. We'll see. It's not really a negotiation. I'm just going to keep going until I get to the end.
We can do it in 2 hours.
This is amazing.
This is the cell cycle. Just so you guys know, this is not going to be on the exam, or I'm sorry, the test coming up Monday. This is going to be on the following one. All right, so the cell cycle. It's all about making daughter cells. We're going to get to that. Not this lecture. We're getting to mitochondrial DNA in the next lecture. Life moves in cycles. If you recall, a long, long time ago, way at the beginning of the course, we talked about frogs. And we talked about killing frogs, slicing them open, doing all sorts of things to frogs in order to figure out why they have too many legs. Do you guys know the life cycle of the frog?
Yes.
Okay, what's the life cycle of the frog, basically? But mainly because it's right here on the slide. Yeah, okay, cool. Hey, good powers of observation there, guys. You've got the life cycle of the frog. You've got an adult frog lays eggs. Those eggs become a tadpole. The tadpole becomes a froglet. That loses its tail and becomes a full-fledged adult frog. And the cycle begins again. All of life moves in cycles. This is reflected in the cell theory. Now, there were 3 tenets of the cell theory. Do you guys remember the 3? Well, give me one of the tenets of cell theory.
All cells come from
Yeah, that's a pretty good one. Omni cellulae e cellulae. All cells come from cells. What else? Cells are the smallest unit of life. And one more? That's this one. Also, okay, so far, all cells come from other cells. Cells are the smallest unit of life. And what are you made of? That's right. All living organisms are made of one or more cell. So today we're really focusing in on this. All cells come from other cells that Rudolf Virchow proposed. Since the beginning of life, about 4 billion years ago, every cell has come from another cell. And those cells just had maybe slight changes from one generation to the next. You haven't changed too terribly much from your parents. I know you hate to hear that. You are, in fact, a lot like your parents. Sorry. You've got 1 head. You've got 2 arms. You've got blood. You've got the 2 lungs. That's every human, but we're not really that different. In fact, we're not really that different from animals either. Pigs and us, we look pretty much alike. Slight differences in snout, but on the insides, so they're tetrapods just like we are. We got 4 limbs. They got 4 limbs. Have you ever had a really late night? In order to reproduce, those cells have to undergo what's called cellular division. This is super highly regulated. You have to know what's going on around you. It's got to be precisely timed. You got to know when you're going to reproduce. For you guys, humans, when is the right time to reproduce for us? When would we consider reproduction? When you hit that's when you're definitely considering it when you hit puberty. That consumes a lot of your considering time. When would you actually, and I take that back. When you hit puberty, are you considering reproduction or are you considering mating?
You're considering practicing.
Yeah, practicing. You're considering mating. Nobody's like, I'm 12, time for a kid. It's not like it was 300 years ago. Parents are forcing you into it. What is? Early 20s is the best time biologically. But my question is, how about you guys? Any of you guys have kids?
When you're established.
When you have enough money to
You have a dog. Okay. All right, let's talk about your pets then. When was the good time to buy a dog? When did you decide to get a dog? You found No, your dog is a special dog. Yeah, but she's really special. What? Not just your age. When you didn't just like one day wake up and go, you know what? That's it. I'm getting a dog.
I'm always choosing them
Why?
because they're cute. I like them.
They're cute, like them.
I want a baby. I have a baby now.
You have a baby now? Wait, you said you want a baby?
No, I'm talking about baby dog.
Right. Okay.
No, I don't have a
Hmm.
Usually it's like late.
baby. My baby.
Late 20s? Why? Why late 20s?
You're up.
What? Because you're focused on your career. Why are you focused on your career? Because what do you need?
You need money to have friends.
That's right. You need money to have kids. You need resources to have a kid.
Societal, pressures and whatnot.
Plus,
Sooner the better. All right. So then you're deciding for yourself. I'm getting old. My cells are breaking down. I'm not going to survive this. There's health reasons. There's resource reasons. Let's say, I don't know. Would you guys want to have a child in a war-torn environment? No. Why? It'd probably die. Not only would it die, you'd probably go with it. I mean, you've all seen those movies. The kid starts crying and then they find everybody. Yep. Don't have that. So, if the environment's not safe, you don't want to reproduce. If you don't have the resources, you don't want to reproduce. Cells are looking around them the same way. They're thinking, and they're not thinking because they're cells, but they're examining the environment. How do they interact with their environment? How do cells know what's going on outside them? What part of the cell interacts with the external environment? The membrane. You've got all these receptors on the membrane. So they're using their membrane to see what's going on around them. They're using internal signaling to figure out, do they have the resources to reproduce? All right, the big component that needs to get transferred from one cell to the other is the chromosomes. Where are the chromosomes located? They are made of DNA, but what organelle are they located within? The nucleus. Now you have to remember, chromosomes, like you said, are really just really tightly bound up DNA. And what is DNA? It's a nucleic acid, and what do nucleic acids do? They dissolve things. No, they're not that kind of acid. And you're going to have to be loud because I'm wearing a freaking helmet on my head, and I can't hear anything. I know. So, what is the point of a nucleic acid? The function of nucleic acid? Doesn't break anything down. Say that again. It stores information. That's just hard to hear. They store information. They store the information on how to make all the bits and pieces that the cell needs. Yes, which is exactly what she said, so thanks. But yeah, you got it too. Well done. So in order to copy these chromosomes, in order to copy this whole set of information, what a cell needs to do is organize them. So during mitosis, the cell will start this division process. The chromosomes thicken and tighten up, so they're easy to move around. When you guys are packing to go on a trip, you've got a lot of clothes, I assume. How do you get them easy to transfer? You fold them. You jam them in a suitcase, right? You get them really packed as tight as you can. How many of you guys have...
You stretch and roll.
You stretch and roll. Okay, lots of different ways of doing this, but let's say you're going on a plane, and you've got a definite, size limit. You squeeze as much as possible into that, don't you?
Wear some of it.
No.
You wear some of it. That's right. Like 8 layers.
They have a weight limit more than a space limit.
They have huge space limits now. Not huge, but they're tiny space limits.
Well, right, but I mean, I can fit twice as much weight as my bag's allowed to be in that amount of space.
That's true. The idea is you have to pack it in tight in order to move it efficiently from one place to another. The cell is doing the same thing with the chromosomes. It's packing it in tight. Now, every single cell out there has a set of chromosomes to it. And all species have a specific number of chromosomes. Now, that's not to say you're not going to have duplications of the numbers. Our species has 23 unique chromosomes, 46 total chromosomes. Frogs, they've got 11 unique chromosomes for a total of 22. Flies have 4. There are other organisms out there that have 4 unique chromosomes or a total of 8. It's not just us. But the big thing to note is they are, in fact, inherited in sets. Here's our chromosomes. We've got 23 of them. You'll notice these oddball ones at the end, the X and the Y. Those are your sex chromosomes. They are arranged in general in order. This picture is called a karyotype. It helps geneticists figure out what really went wrong with you. All of it. Those weird oddball 2 at the end are called the sex chromosomes. Typically, for humans, they're denoted as X and Y. The X chromosome is the big one. It's filled with lots and lots of genes. The Y chromosome is little tiny leftover genetic crap. Because that's really what it is. Yes? A haplogroup? A haplogroup is going to be a... it's a group of genes that are traveling together on a chromosome, yeah. So it's nothing more than that. It's just, you would be put into a haplogroup with other people whose genes in that region are just like yours. So it's a group of genes moving. Yeah, that's how they trace ancestry. So if you're male, you got an X and a Y. If you're female, you got an X and an X. All of the rest of the chromosomes are termed autosomes. Anything not a sex chromosome is an autosome. When a cell has 2 complete sets, 2 full sets of chromosomes, it's termed diploid. Most eukaryotic organisms are diploid. You are a diploid. You have 23 unique, 46 total. So here's haploid, N, diploid, 2N. And those N's mean something. How many of you guys have taken algebra? Yay! We're back to that. N is just a variable. It's like X. In this case, the N number is 3. So what's 2N equal? 6. Just that easy. If the N number was 5, what's 2N? 10.
Oh, okay.
If the N was 10, what's 2N? 20. Oh, now you're getting there. What if N is 30? What's 2N? Awesome, we're there. Most organisms that we're dealing with are diploid, 2N. If you go into bacteriology, if you go into microbiology, totally different story. Some cells in your body are gametes. Gametes are termed haploid. They have half the number of chromosomes. Your sperm or your eggs are going to be considered haploid. And the reason for that is, do you guys know how fertilization works? Making sure. You've got a sperm and an egg. The sperm fertilizes the egg, and then you go from N plus N gives you what? 2N. So that's how you end up with your diploid number. But let's just talk about, we're not going to deal with the gametes until a little bit later in the lecture. We're going to talk about eukaryotic cell division, where we inherit these chromosomes in complete sets. For you, the diploid organism, you are going to get a pair of each chromosome. What's the gender of this person?
Is there an X and a Y, or is it just...
It's a boy. Why is it a boy? X and a Y. When you inherit these pairs, if those pairs look very, very similar to each other, they contain the exact same coding regions, they are termed homologs, or homologous pairs. Important term to know. So if I was to have one chromosome, and it had circle, triangle, square, in another chromosome, it would still have a circle, but it might be slightly different. They're encoding for about the same stuff. There we go. They're encoding for the same stuff. Those are homologous pairs. Where'd these come from? Well, you get one from your mom, you get one from your dad. And your mom and dad really aren't too terribly biologically different from each other. Pardon? Yeah, surprise. On these chromosomes, you have a place that the gene will be located. Okay, I'm throwing a lot of terms at you. Guys, we're hitting the part of the course where this is my language, and I will switch into biological speak. I will switch into terms that you may not understand because, to me, they're the easiest way to describe a concept. If I use a term you don't understand, it's because I think for some reason I've already taught it to you. So, to start with, a gene. A gene is a polypeptide encoding region. Polypeptide's a series of amino acids, right? It has the information to build one polypeptide. And it's found on a chromosome. These genes are DNA sequences found on a chromosome. Where they're located on the chromosome is called the gene locus, the spot. Sometimes, when you have a homologous pair, those genes are exactly the same. There's no difference between them at all. Sometimes, they encode for basically the same thing, but there are slight differences. If there are slight differences, they're called alleles. So that's a lot of terminology I need you to know there. Gene, homolog or homologous pair, gene locus, and alleles. Sure, I'll give you a second to write that down if you haven't done so. What?
Week 11.
With 11, chapter 11? Oh, I have no idea. This is the lecture is in with right after photosynthesis. Should be under energy transfer. We good?
The name of this PowerPoint is
The cell cycle. So, one of your homologs comes from your mom, one of them comes from your dad, they come together, they form the body plan to make you. It gets passed on to their offspring. When we're talking about something from your mom, we say maternal. When we talk about something coming from your dad, we say paternal. Like paternity test, checking for the father. Or patricide, you know, just in case. The vast majority of the homologous pairs differ by less than 1% between chromosomes. There's less than 1% difference in that gene structure between any two homologs. That really goes to say how alike we are to each other. Sure, we may look different to us, but the thing is, we're really, really, really similar. More than 99% similar to each other. Oh, the exception to that, of course, is the X and Y chromosome. The X and Y chromosome are so different in size that they're really not terribly alike. When you're studying the movement of these chromosomes, you're studying cytogenetics. That is, the chromosome moving during cell division. You can see these things under the microscope. To study it, they look at karyotypes. Karyotypes are just pictures of the chromosomes that are sort of cut out and rearranged. Back when they first started doing this, 50s and 60s, literally they would take a picture with a camera, print out the film, and then take a pair of scissors and clip out the chromosomes and line them up on a paper. Then they look at them, see where are there differences between the different chromosomes. Now we do pretty much the same thing, except instead of printing it out, we do it all on the screen. Question? No? Okay. Whatever. So, take a look at this. Is this a boy or a girl? Girl, how can you tell? Now, is this girl totally normal? No, why? Something's jacked up somewhere.
She's got, there are 3 in 21.
There are 3 chromosome 21s. This is known as trisomy, specifically trisomy 21. This is what causes Down syndrome. Now, taking a look, what are chromosomes made of?
Peptides.
DNA, and wrapped around peptides, you're right. DNA wrapped around histones. DNA is composed of a whole bunch of genes, and genes are polypeptide encoding regions, right? Here we've got 3 of them. What we've done is we have an instruction manual that's got extra pages. So we're producing too many proteins at this point, too many polypeptides. And that leads to toxicity in the cell, leading to various identifiable characteristics of the organism. How about this, boy or girl? What? Boy, why? It's got an X and a Y, but it's got 2 X's. It's not hermaphroditic. This is actually, I believe it's going to end up being, I want to say female, but with underdeveloped genitalia. Still, you've got another issue. You've got an issue with the chromosomes. Too many proteins being produced. What's wrong with this one? Oh, wait, first off, numbers or letters are underneath. So this is X and Y. So this is a boy or a girl? It's a boy. Totally normal? No. What's wrong? Look at number 9. You're missing an entire piece of instruction. That's going to cause some issues. There are deleted sections of the instruction manual. And you said 1 and 3. 1 and 3 have actually switched pieces. That's not going to cause a problem in this organism because all the instructions are there. But it will cause a problem in their offspring.
So that's the effect of inbreeding on like the royalty levels over the years.
This actually has nothing to do with that. That we're going to get into in the next lecture. As far as inbreeding goes, this is actually a lot of times just caused by random chance. That is, yes, that's one reason different traits will skip generations.
What like mainly causes it?
Say that again?
What like mainly causes it, or just the action?
This is going to end up being caused by a lot of different things, one of which is age. So how old you are when you reproduce. No. Like if you're 36, this is what happens.
The younger the better.
Yeah, the chances are lower. If you have if you're older, if you're like in your late 30s, early 40s, you're termed a geriatric pregnancy. But that's why, because of this.
It's only like a 1 to 3% chance.
Right. All right, so this is an actual karyotype, boy or girl? It's a boy. It's a boy. It's got an X and a Y. Are there any issues?
Yeah, with Mars.
Yeah, okay. This is it was done in March. Number 6. 6 is missing an entire chromosome. Anything else? 13. 13's missing a top. What else? 5, missing a bottom.
17's kind of funky.
17's kind of funky. 2's missing a top. So this organism started growing. The egg fertilized the sperm, but within the first trimester, it ended up spontaneously aborting. The organism couldn't survive. It couldn't produce enough proteins. So they found out they were pregnant, and then shortly after, they weren't pregnant anymore.
You think that was just because of 6, or that has something to do with multiple?
It's going to be probably 6. If you end up missing any of like the top 15, you can't survive. There's just too much instruction manual you're missing. So how do we get there? How do we get to look at these chromosomes, look at all these different conditions? It all comes down to the cell cycle. Cells have a life, just like you do. And in their life, they have to grow, they have to reproduce, they have to make sure they're surviving. So there are 4 steps to it: G1, S, G2, and M. So G1 is the first gap. This is where the cell's growing. It's eating. It's checking out its environment. S phase is where it's called synthesis. That's where the cell is going to copy its DNA. G2 is the second gap. It's checking itself out. It's making sure it's ready to reproduce. And then M is the mitotic phase. I'm pretty sure most of you have taken biology at some point in your life. You learned about mitosis. And then you probably heard like interphase is a part of mitosis. Yeah, actually interphase is most of it. Like the vast majority of the cell's life cycle is not reproducing, just like you. You don't spend all of your life not trying to reproduce. You might spend all your life trying to reproduce. You don't spend your whole life actually in the act of reproduction. You take time out to do things like eat, rest, recover, grow bigger. The first of this phase is G1, S, and G2 are called interphase. Takes up the vast majority of the cell's life. This is when the cell grows, it copies its DNA, it checks itself out before it tries to divide. The first phase of it, the first phase of interphase, is called G1, growth. All the cell's doing is eating, getting bigger, and bigger and bigger. It's followed by S phase, or synthesis phase. During S phase, the chromosomes all get replicated. They get copied. Here, how many chromosomes are in this cell? 2. So you can count chromosomes by counting the middle of them, the centromere, that middle part. So there are 2 here. How many chromosomes are here? 2. Well, I just said, during this phase, you're copying your chromosomes. If you look, you've got half as much genetic material here than you do at the end. Or to put it another way, at the end, you've got twice as much DNA.
How you did that?
It's amazing. We should go through a whole process. This is it occurs through a process called replication. We'll talk specifically about the molecular mechanisms of replication toward the end of the semester. After that chromatid, that chromosome is copied, it forms 2 sister chromatids joined together at a centromere. Lots of terminology. So here we have 1 chromosome made of 2 sister chromatids joined at a centromere. So when you hit the end of S phase, you have twice as much genetic material. 2 chromosomes at the top, 2 chromosomes at the bottom. The difference is here we got 2 sister chromatids joined at a centromere. Any questions thus far? Once we get through synthesis, S phase, we enter into G2. During G2, the cell begins to produce proteins it needs to reproduce. It starts churning them out like crazy. It begins to double check the DNA, making sure it's all put together well. This is where the cell sort of has an opt-out point. This is a phase that may or may not happen. It's called G0. It's not called go. It's like the opposite of go. It's G0. This is where the cell stops all cell division. If the cell's not big enough in G1, it doesn't divide. It enters into a G0 state, a suspended state. If the DNA has problems with it. During G2, you check out the DNA, and if there's a problem with the DNA, the cell stops dividing. There's another phase. If there's a problem in mitosis, the cell recognizes the problem and stops dividing. So you got G1, S phase, G2. And if there's an issue with any of them, the cell enters into G0. That's interphase, and is the vast majority of the cell's life. M phase, mitotic phase, occurs after interphase completes. And it's made up of two parts, mitosis and cytokinesis. Hey, so you know that whole issue I've got with mitochondria? Here's another issue I've got. So, again, when you were younger, you learned about mitosis, right? And people always said mitosis is cell division. Yeah, it's not. Mitosis is the division of the nucleus. Cytokinesis is the division of the cell. And they are two very distinct processes. So with mitosis, we are dividing the nucleus. So we went through this S phase process. We've doubled the amount of genetic material. What mitosis does is brings it in half again, so that by the end you have an identical daughter nuclei. So S phase, we double the amount of genetic material we have. Mitosis, we have it, and thus we're right back where we started. So the offspring has the same number of chromosomes as its parent. Cytokinesis, on the other hand, is the division of the cytoplasm and all of the organelles. It splits them up into the daughter cells. For a 24-hour dividing cell, and I want to make it clear, most cells do not divide in 24 hours, but I thought I'd break it down into what your day was like. You've got G1 phase. G1 phase will last about 11 of those hours. This is where you're looking for food. Welcome to college, right? That's like half your day, isn't it? You're like, man, I'm hungry. I should eat something. Probably half of you are thinking that right now, except Sarah, who ate an apple. Some of you guys are like, sneak the food. So most of the cell's life is actually concerned with find food, grow bigger. The next part is S phase. S phase is where you're copying that DNA, and that takes a fairly long time. About 8 hours, a third of the cell's life cycle. Getting ready to reproduce. G2 phase is checking itself out before it reproduces, about 4 hours. And the actual reproductive part, that mitotic part, takes about an hour. 1/24 of its life. If you were to, I don't know, get ready to go out on a date, you don't spend the entire, I hope you don't anyway, you don't spend the entire day just worrying about that date. Well, you may be worrying about it, but you're doing other things too. You're eating, you're going to work, you're doing other things. That's G1. Then you start really worrying about that date. You start prepping for it. You take a shower, I hope. You brush your teeth like 6 times, just in case. After that, you look at yourself in the mirror. You're like, am I okay enough? Does this work? That takes some time. The actual reproductive part, or the part where you get to the end of the date, that's really short.
You know a lot about it.
No, I really don't. It's unfortunate. This cell cycle must be heavily regulated. And the way it's regulated is through some proteins called cyclins. You've got cyclin A, B, where's cyclin C? So cyclin D and cyclin E. What I want you to notice just about this pie graph is as cyclin A increases, you're starting off at G1, right? G1, you have cyclin E increasing. It peaks. When you've got a certain threshold of cyclin E, cyclin A kicks on and starts to grow, and that causes cyclin E to diminish. What we're seeing here is biochemical pathway controls. As cyclin A peaks, cyclin B kicks on. And then as cyclin B peaks, you hit the beginning of mitosis. The reduction of cyclin A and the increase of cyclin B cause mitosis to occur, and then we begin again with cyclin D. It's even easier to look at with this graph. You got cyclin D rising, and as cyclin D rises, cyclin E kicks on. As cyclin E peaks, cyclin A kicks on. As cyclin A peaks, cyclin B kicks on, and they all end in mitosis. They're all gone by then. And you guys are really getting good enough at reading graphs that you didn't even notice that this was a graph that was written totally in German. So that's kind of nice. It's not like they just spelled concentration wrong. During mitosis, you have to hit these checkpoints. You have a G1 checkpoint, a G2 checkpoint, and a mitotic checkpoint, or metaphase checkpoint. During G1, you're looking around. You're using those proteins in the membrane. You're saying, are the conditions favorable? Is there enough food around? Do I have enough space to divide? We get to G2, and we get another batch of proteins. They're primarily concerned with, how's the DNA doing? How's this, the instructions? Do I need to revise anything? And then during the third checkpoint, we're checking to see if the chromosomes are connected and ready to be pulled apart. If any of these checkpoint fails, there are critical errors that will occur for the human. There are consequences. If the cell does not enter into G0, when it is broken, that is when you get tumors. That's when cancer happens. Cancer is entirely a genetic condition. If it doesn't enter into G0 when it's supposed to. It happens to cells that are broken. If the cell doesn't pass the checkpoints, most cells are going to hit that checkpoint and be like, everything's fine. But if a cell hits that checkpoint, and it's broken cell, but it just blows through the checkpoint, that's when you can get problems. If you have a mutation in your checkpoint genes, and suddenly the cell's able to divide rapidly without regulation, that's how tumors grow. This tumor was taken out of a woman. It's roughly the size of a watermelon. It was taken out of, I think it was taken out of her thigh. I know. She didn't even realize it was there.
It was a watermelon.
She was a large woman. Part of the reason she was so large was she was filled with tumors. No, she didn't die. This is a totally benign tumor. What tumors end up doing, benign tumors anyway, is they're going to just take energy and nutrients from the rest of your body and use it to power these cells that won't stop reproducing. Of course, you have malignant tumors. Tumors that spread and grow and get bigger and bigger and bigger and continue to spread. So in the end, if you don't have controls on this cell cycle, that's where cancer comes around. Downside is when we learn about how genetics works, it looks like if you live long enough, likely you're going to get cancer. Yeah. Yeah, no, that's the way it works. Because of the way genes get shorter and shorter and shorter every time they divide, as cell divides. Yes, there are definitely environmental conditions that will impact tumor formation. But there's also a strong inheritance pattern to it. What? Oh, you did? No, it's cool. I'm good. All right, so we control how our cell divides. Then we get into, after we've gotten through G1, S, G2, we're into mitotic cell division. Where we're going to take that nucleus and divide it in half. It's going to be genetically identical to the parent. That's also known as asexual reproduction because it's done without sex. When a chromosome begins to divide, you've got these two identical chromatids. When does this happen? What part of interphase? When you get two identical chromatids. We're not in prophase yet. I'm just talking about during those three phases, G1, S, G2. G2, no. S phase, synthesis phase. That's when they're copied. They get joined together at a centromere. In order to organize these, you start to form what's called a mitotic spindle. This is a bunch of microtubules. Where have we heard of microtubules before? In the cell, yeah. Yeah, they're the long things that go from one point of the cell to the other. What do we use them for? Transportation, moving things around. The railway system of the cell. So the microtubules are going to grow out of these microtubule organizing regions, the centrioles, the centrosomes. They're going to expand outward and connect to where the centromere is, the center of those chromosomes. The problem is, chemically, they cannot connect to a centromere. So instead, they connect to a belt surrounding the centromere called the kinetochore. Have you ever pulled somebody around by their belt? Yes. Because that's the way you control where they go. If you pull them by the belt, they have to go where you pull them. Right? Center of gravity. You guys have done that to somebody, I assume. You grab them by the belt and you pull, and they follow because they can't not follow or they'll fall over. Kids with a leash? It's not, no, because if you've held kid with a leash? No, see, if you pull on them, they just pull back. Yeah, but the person with the belt, you pull them, they have to go. They need to redesign those leashes. Yeah. That belt is called the kinetochore. All right, so we got all our pieces. Now we're going to go through the steps. This is the part where you guys are like, oh, God, I remember this. If you don't remember it, great. It'll be easier to fix all your misconceptions. All right, so we've got several steps to mitosis. Typically, most people will start off with interphase. Well, interphase is not actually a part of mitosis. It is the vast majority of the cell's life cycle. But then you have prophase, prometaphase, metaphase, anaphase, and telophase. And during that process of mitosis, cytokinesis is occurring. I am going to have you describe not only the steps of interphase, I'm going to also have you describe the steps of mitosis. It's going to be an essay question. We have to list them and describe them. Prophase, prometaphase, metaphase, anaphase, telophase. Of course, you can have a mnemonic to remember that. It's the PP mat. Yep. I got a fun story for you. Yeah, I know, right? So I have stupid friends. I'm sure you do as well. Well, one of my male friends decided the shower goes to the same place as the toilet.
Oh, why not, right?
Right, so it would save time if he just peed in the shower while he was taking one. And he didn't tell anybody about this when he was living with them. And what he did was he ended up taking, you know those shower mats? Yeah. That are in the shower, not the ones that are outside. And so he would just aim at that. So he called it the pee-pee mat. And that is, honest to goodness, how I remember the steps of mitosis.
How long did it take?
For him to tell us that? It took probably about 3 months, and it only came out randomly. He's like, yeah, I pee in the shower. We're like, what's wrong with you? That's not okay. And he honestly, though, spent like 20 minutes trying to describe how he saved time.
Yeah, like 10 seconds.
I know.
That's not even 10 seconds.
All right, so interphase. We got G1, S, G2. We grow. We replicate our chromosomes. We check out the area around us. Then we enter into prophase. Are you guys ready for this? I got pictures and descriptions. I'll talk you through it. During prophase, the nuclear envelope begins to dissolve. It breaks into vesicles. the mitotic spindle begins to form at the centrosomes. They begin to form a lot of microtubules coming out of them. They start to move to either end of the cell. And the chromosomes start to get thicker and thicker and thicker so you can see them. Right, so the nuclear envelope dissolves, the mitotic spindle forms, the chromosomes condense, they get thicker. Then we enter into, do I need to go slower? Do I need to go through that again? No? Just move, move. Get this over with.
I just feel like this stuff is going to be on Monday.
Pardon?
Like, this is the stuff I'm going to remember on Monday.
This is the stuff, I know, I'm sorry.
On Monday.
I'm going to study on Saturday.
In prometaphase. Prometaphase comes so first, or before metaphase. The microtubules and the microtubule organizing regions, the centrosomes, move to either end of the cell, and they connect to the chromosomes. That's the whole point of this phase. Connect the microtubule to the kinetochore, surrounding the centromere on the chromosome. So step 1, prophase, condense the chromosomes, get rid of that nuclear membrane, and start to form your mitotic spindle. Step 2, prometaphase, you're going to attach the microtubule to the chromosome. Step 3, metaphase. Meta means middle. Not just because this is the middle phase, but because the chromosomes literally get lined up along the middle. The microtubules push or pull them into place. It's like if you had two fishermen who caught the same log, because really they didn't catch a fish. So you've got this log on a hook. Based on where they are, they can reel in their they can reel in the log, or the other person can reel it in. And that back and forth pull will allow them to position the log wherever they want between the boats. So now we've got all the chromosomes lined up along the middle. Next, we have anaphase. In anaphase, the chromosomes get pulled apart to either end of the cell. And then telophase, the chromosomes start to decondense, the nuclear envelope reforms at both ends of the cell, and the cell will end up with two distinct nuclei. All right, so first step, prophase. Chromosomes thicken, nuclear envelope dissolves, the centrosomes begin to produce microtubules. Second phase, prometaphase. The microtubules bind to the chromosomes. Third phase, metaphase. The chromosomes are going to line up along the center of the cell. Then we have anaphase, where the chromosomes get pulled to either end of the cell. And then you have telophase, where the nuclear envelope reforms, and the cell has two distinct nuclei. We have not split the cell. That comes with cytokinesis. In cytokinesis, you get a cleavage furrow that forms. You get a belt that goes all the way around the cell, and that belt's made up of actin filaments. And what does actin do? Actin acts. Actin causes actions in the membrane. So that belt gets tighter and tighter and tighter until the cell pops apart. It's just like making a giant vesicle in the Golgi, or in the endoplasmic reticulum. It's like a really bad Saw movie. If you pull a belt tighter and tighter and tighter and tighter, eventually that cell will split in two. Now that's how it works in animal cells, but in plant cells that can't happen. Plant cells and animal cells are different because of what? Chlorophyll, okay, but also a cell wall.
I got that right.
You did get that. Well done, well done. So you got the cell wall. That cell wall is not going to be able to be split by actin. So instead what happens is the Golgi apparatus ships a whole bunch of proteins to the center of the cell. And it begins to build a whole new cell wall right in the middle. Have you guys ever played in a creek before? A creek, yeah? Okay, just making sure, 'cause some people didn't have a childhood. Yeah. No? Have you ever made a dam in a creek? Yeah? How'd you make the dam? Rocks. So you take one rock, you put it in the creek. Water still goes around it. It's not an issue. 2 rocks, 3 rocks, 4 rocks. As you pile more and more rocks, your dam becomes more solid. So what's happening is, in the center, it's dropping off protein after protein after protein. At first, it doesn't do much. But over time, it builds up. Until eventually, you have a solid wall called a cell plate that goes from one end of the cell to the other, splitting it into 2 distinct daughter cells. And then what happens? They're split apart and they can live their own lives.
This new cell wall divide itself in half down the middle when they split.
Yeah, well, now it's, oh, like right here? Yeah, that's an actual split between them. And plant cells, like we talked about with plasmodesma, that's how they're going to get formed. They're going to be built right into that. So, the point of all of this is to ensure there's genetic consistency from one generation to the next. So that one organism, the parent, looks a lot like the offspring. Because if there's not that genetic consistency, then you're not going to be able to survive as well. This is especially important in multicellular organisms. It's really important for your body cells to make exact copies of themselves, because if the instructions were different, your cells wouldn't recognize each other. Do you guys know what these are?
Yeah.
What are they? Armadillos.
Armored ones.
Yeah, I know, they're little armored rats, right?
Well, I don't know, rats are more like, I don't know,
They're kind of like armored rats.
anteaters.
Yeah, that's true. With an armadillo, the reason I put them up here is because did you guys know that armadillos are always born as quadruplets? Every single armadillo is a quadruplet. Isn't that crazy? So the takeaway message is they have that genetic consistency between them. And if you ever run over one with your car, don't worry, there are 3 more just like it out there. So I'm going to take a quick break at this point. Come back in, what, 5? All right, so for the rest of the course, we're going to talk, or for the rest of the course, for the rest of the day, not the rest of the course. We could do a whole course on this, but no. We're going to talk about meiosis, which really thanks, glad you're here. Which really gets into the nitty-gritty of how we came about as individuals. It's all about sexual reproduction. We're going to have 2 haploid gametes coming together to unite to fuse to form a diploid zygote. Now, the way this works in humans, and I just want to put this broadly out there, the male's producing sperm, the female's producing eggs, the sperm and the eggs come together. It's not just magic, it doesn't just happen like randomly.
Happen in your sleep.
Right. And you form a diploid zygote. The reason I have to explain that is because I didn't realize it, but apparently there's some people out there at the college level who really didn't know. I had to, I said, guys, all right, so sexual reproduction, you all know how it works. We're going on, and they're like, egg and sperm, and I was like, yeah, they were homeschooled. Their parents were really, no, I wouldn't say conservative. They were just very reluctant and embarrassed to talk about the way sex works.
Okay.
It's okay, my parents were too.
See, the town where I grew up had a very, very bad health program.
So, the way this works, haploid, they fuse. This becomes known as a zygote. That zygote then is going to very quickly undergo mitotic cell division. Okay. As you are developing, I'll just put this up here because it's not in the notes, but you know, I'll do it anyway. You've got a zygote. That zygote's going to split in half to create two 2n cells. Those split in half to create 4 2n cells. Those split in half, and how many have you produced? Keep going. 8. 8 go to 16. 16 to 32. 32 to 64. 64 to 128. 128 to 256. 256 to 512. 512 to 1024. And so on. Eventually, you have enough cells to go from the single zygote to what's known as a blastula, which is a hollow ball of cells. Blastula. Not really. It's got a different root. That blastula, this hollow ball, is going to what's called invaginate. It gets an indentation in it. It's almost like taking a balloon and shoving shoving your fist into it. It forms a concavity around your fist. That blastula then has undergone gastrulation and become a gastrula. This is where you get your skin, your digestive system, and all of your internal organs. That's development. In order to get there, though, you needed to have an egg and a sperm. So we get those egg and those sperm through this process called meiosis. So to produce these gametes, we have to first go through G1, S, and G2. Where have we heard of this before? You're right, it's mitosis. It's pretty much the same stuff. There are a few slight differences. In mitosis, during prophase, you get your homologous pairs have formed and they've thickened. During meiosis, you take those sister chromatids and those homologous pairs and they come together to form a tetrad. So instead of having, time for a puppet show, you've got chromosomes. So these chromosomes look a lot alike. During S phase in mitosis, they replicate. So now you have 2 sister chromatids joined together at a centromere. Does that make sense? They're going to line up at the center of the cell during metaphase and then split apart. In meiosis, it's slightly different because what happens is you start off with 2 chromosomes. They undergo synthesis. They copy each other. But instead of staying apart, they come together to form a 4-chromatid module called a tetrad. Or synapsid, if you want. Those synapsids, those tetrads, allow us to undergo a process called crossing over. Crossing over is where there's a physical exchange of chromosomal elements. Here you've got the tetrad formed. These legs are so close together and so chemically similar, when they cross, they can actually switch spots. So you've got your dad's is red, your mom's is green. They're able to swap pieces.
And now you're part of your father's body?
Now you're mixed up, yeah. It's called a recombinant chromosome. Or recombinant chromosome. They've recombined.
Oh, okay.
It's the equivalent. You guys ever run a 3-legged race?
Yeah.
And there's a few ways of doing that. One is, you tie yourself to another person's leg and you sort of like run along together. But what inevitably happens is one person just picks up the other and drags them because you have that one ultra competitive person.
Yeah.
Yeah, okay. As long as we're there. Slow down, slow down. Oh, my leg. It'd be like at the end of the 3-legged race if your leg and their leg switch places. Yeah, they're similar enough. You got a knee, they got a knee. You got ankles, they've got ankles. You undo the tie and suddenly you're wearing their shoes. Yeah, well, thanks for pointing out the flaw. Cool. Where these things cross over, where these sister chromatids cross over, you have what's termed a chiasma. Right, because it's named after the Greek letter chi. Looks like an X. The arms of one chromosome cross over the other. So here you've got A, B, C, D, E, F, G, H, I, J, K, L. Here, same thing. Here, lowercase, here, lowercase. Where they cross, now you're going to get a recombination event, a crossing over. So you have the first one stays the same. The second one becomes A, B, C, D, E, F, G, H, I, little j, little k, little l. Then little a, little b, little c, little d, little e, little f, little g, little h, little I, j, k, l. This is where we get variation. This is what allows us to be different from one generation to the next. Regulation of crossing over is highly, highly regulated. In humans, it only really happens about once, or maybe twice in a gamete. In plants, it could happen like 20 times. Plants use this as a major way that they look different from each other. So here we've got chromosome ACB, little a, little c, little b. There's a double crossover event, so we get big A, little c, big B, little a, big C, little b. Slight differences. So that when each of these goes into their own gamete, you've got one gamete that's got chromosome ACB, one that has A, little c, B, little a, big C, B, little a, little c, little b. All unique. So in order to get these unique chromosomes that will go on to make unique people, you need to undergo meiosis. There are two parts to meiosis. Meiosis 1 and meiosis 2. Meiosis 1 is composed of prophase 1, metaphase 1, anaphase 1, and telophase 1. All of which... Pardon? Yes, phases of meiosis. First phase, prophase 1. The chromosomes thicken, the tetrads form, crossing over is occurring. The nuclear envelope dissolves, the mitotic spindle forms. Metaphase. The microtubules have attached to the tetrads and pulled them to the center of the cell. Anaphase, the tetrads are pulled apart, moved to either end of the cell. And telophase, the nuclear envelope reforms, and you get two distinct daughter nuclei. So to recap, prophase 1, the nuclear envelope dissolves, the tetrads thicken, crossing over occurs. Metaphase, the microtubules push or pull the tetrads into the center of the cell. Anaphase, the tetrads get pulled apart. And then telophase, the nuclear envelope reforms to give you two distinct daughter nuclei. Question? That's the end of meiosis 1. Meiosis 2 will occur next. Do you guys feel comfortable with mitosis? Because here's the deal. Meiosis 2 is exactly like mitosis. The only difference, the huge difference, is that meiosis 2 does not have S phase. Yeah, so they'll just have G1, G2. Yeah, there's no S phase in between. If there's no S phase, what occurs during S phase? Synthesis, chromosome replication, which means we are now, by the end of it, having the amount of genetic material that we had previously. So you got meiosis 1, where you got those tetrads and crossing over happening. And then you've got meiosis 2, where you've got effectively the same thing as mitosis, except there's no S phase, no copying the chromosomes.
Where does that happen?
Where does that happen?
Yeah, where does meiosis 2 occur?
Meiosis 2 is going to occur, in the cells that are producing the gametes. So, like the spermatophyte in the plant, the oocyte or the oogonia in the plant, primary ogonium in the female, things like that. So not throughout the whole body. So that's meiosis 2. You put meiosis 1 together with meiosis 2, and you end up with a haploid gamete at the end. You'll notice we start off with 2 chromosomes at the top. We end with 2 chromosomes at the bottom. The difference is these chromosomes, these final gametes are all unique. They all have different types of genetic material in them. Different, not types, combinations of genetic material. So for the inevitable question of what's the difference between mitosis and meiosis, mitosis will produce diploid daughter cells. Meiosis will produce haploid daughter cells. Mitosis produces identical daughter cells. Meiosis produces unique daughter cells. Mitosis generally produces body cells. Meiosis produces sex cells. And that's all I got on mitosis and meiosis. Do you have questions on them? Okay. Concerns? Otherwise, I want to talk about just chromosomes themselves. Variations in chromosomes. There are 3 ways to get variation in chromosomes, the big structures. You can get a mutation that alters the structure of one chromosome. You get a mutation that causes variation in the number of individual chromosomes. Or you can have mutations that lead to variations in the number of chromosome sets. So first off, mutations that affect a single chromosome. You could have deletions, duplications, inversions, or translocations on a single chromosome. First up, we have deletions. What does it sound like? Deletion. Take something away, delete it. So in this case, if we have A, B, C, D, E is normal, a deletion would have A, B, D, E. You've taken a piece out, and you've removed it. It's gone now. You no longer have those instructions. This leads to damage in the cells because you don't have the necessary information to make proteins. You could have duplication, doubling. For duplication, a piece of the chromosome is pulled out, copied again, and put back in. Well, this leads to problems because now there's double the amount of genetic material present. You could have an inversion. Do you know what it means to invert something? Yeah, turn it upside down, or flip it, inside out, flip it. So here what you're doing is you're taking a piece of the chromosome, pulling it out, and flipping it around. Now this typically does not lead to many problems, except right on the edges, right on the points where it comes back in. Those genes might be disrupted. Why does it do it? Right, no. So inversions typically occur because of a loop that occurs during DNA replication. It's a bad thing. It's not a good thing. So it's an accident. Typically, the cell is looking for these accidents when it's going through G2 phase. But if it doesn't catch them, this is what happens. There are 2 kinds of translocation. A simple translocation, where a translocate means to move across. So you're going to have a piece break off of one chromosome and attach to another. That's simple. Pops off, attaches over there. Versus reciprocal. In a reciprocal translocation, the chromosomes are going to both break off a piece and both switch pieces. This typically doesn't lead to much problem in that organism. The reason it doesn't have much of a problem in that organism is because the instructions are still there. So let me ask you some questions here. We got a normal chromosome, A, B, C, D, E, and chromosome X, Y, Z. What kind of chromosomal abnormality is it if I was to have A, B, A, B, C, D, E. What's that? What? Duplication. What if I was to have A, B, D, C, E? What? Nothing's deleted. It's all still there. It's an inversion. It's been pulled out and flipped. So it's as if you have A, B, C, D, E, and the C and the D got pulled out and flipped upside down and then put back in. All right, how about A, B, C, D, E, Y, Z? That's a simple translocation. A piece broke off and moved. But if I was to have A, B, C, D, Z, and X, Y, E, that's a reciprocal translocation. This is going to be matching. Do you guys want more practice with that?
If you have like Down syndrome and the extra gene is deleted, then like corrected.
If you have Down syndrome and the entire extra chromosome was deleted, it would fix that condition, yes. Any questions? Do you want me to go over that some more? No? Okay. So that's if there's a problem on a single chromosome. You might have a variation, a mutation that causes changes in the number of entire chromosome sets. Haploid and diploid are normal ones. Haploid are sex cells, diploid are U, 2N. It's called euploid when it's normal. But sometimes there's a mistake happens. Sometimes the microtubules don't grab onto those chromosomes to pull them apart. And you end up with too many chromosomes. 3N. 3N is termed triploid. Or tetraploid, 4N. Now we're producing way too many proteins, but we're producing them in about the same ratios we were before. So euploid means normal sets of chromosomes, 2N. Polyploid is 3 or more sets of chromosomes. When you make, what happens then? Humans don't tolerate it. It's not Down syndrome yet. That's going to be the next step. Humans don't tolerate this. Animals don't tolerate this. There's no point in fixing it. You're dead. Yeah, it dies very, very early. It doesn't work. Plants, they use this all the time as a means of speciation. The final category, back to Down syndrome, is aneuploidy. Here you have an alteration in the number of a specific chromosome. For Down syndrome, you're termed trisomic because there are 3 chromosomes. Everything else has 2. One of them has 3. So that's trisomy 21. You might be monosomic. What does mono mean? 1. In this case, you only have a single chromosome. In this case, there's a single sex chromosome. So it's monosomy X.
What is Turner syndrome?
What is Turner syndrome? We'll look it up later. In fact, extra credit? Does that sound like a good idea? No, I was actually thinking about having you guys work on looking up some genetic conditions and describing them. No, not for extra credit. Yeah. Yeah, but it wouldn't be graded. Just for fun. You're good with this. So we got Turner syndrome, which is a monosomy. There's too few proteins being produced. So you got too many, and over there you got too few. Animals do not tolerate polyploidy. Few exceptions. There are certain snakes and certain frogs that can be haploid.
It makes you short.
Haploid is half. Is N. Plants, on the other hand, they can be polyploid. They like polyploidy. Polyploidy results in various flower sizes, various fruit sizes. This is a daylily. It's about a 2-inch wide daylily. It's 2N. When I doubled the number of chromosomes in it, it became 4N. And it had a much larger flower, more proteins being produced. It could be. I don't know how they do that. But I will tell you, with strawberries, do you see this strawberry at the top? That's you may have seen those like growing in your yard if you didn't cut the grass fast enough. They're really, really small. They are, people call them snake berries, I think. They're edible, but they're not tasty. They're bad. Yeah. They are really, really small. They are they are diploid. 2N chromosomes. Well, over time, because we're awesome at agriculture, their chromosomes doubled, so they became 4N. And they doubled again. They became octoploid, 8N. And that's where we get our commercial strawberry cultivars. So this polyploidy works really well in plants, not so much in animals. As far as aneuploidy goes, we can tolerate aneuploidy. One too many or one too few chromosomes, as long as it's in the much smaller chromosomes. A lot of genetic conditions come from this, but some other ones can come from just missing a piece or having too many proteins. It could be there's a genetic component to it, because a lot of the problems come from microtubules being able to form right. So aneuploidy happens, and that person's still human. That person has everything else working. They've just got maybe a little bit too many proteins being produced or a little bit too few. I think webbing, too, on their neck, right? Do you have webbed toes and fingers? Not anymore. Did they, like, cut them? Pretty cool. Let's see what happens to your kids. Come back in, like, years. Bring them.
You should have totally kept the web stuff like.
Any questions on this, guys?